But we have, as Thomas was just suggesting, a really nice talk now by Manuel Paina of Axxam. He will be talking about Organella ion channel recordings, which is a really interesting area that we're getting more and more interest upon, and it seems like a number of drug discovery programs are heading that way. I think they're quite challenging targets for obvious reasons, but Manuel Paina is an expert in this area, so he's actually been doing some great work at Axxam, some of it with the Q-patch as well. So I potentially, if it's available, I can hand you over to Manuel and he can talk. And again, please do put your questions into the stage Q&A for Manuel that we can sort of apply to him at the end of his talk. So I will hopefully hand you over now to Manuel if we're ready for Manuel to take over.
Hello.
Hello, Manuel.
Hi to everybody.
Welcome to the stage. You're up.
I'm fine, and I can share my screen. Okay. Hi to everybody. My name is Manuel Paina, and I am principal scientist in Axxam. And today I'd like to share with you the results that we obtained using the Q-patch, but not with the cells, but with the organelles. And the title of my presentation is Journey to the Center of the Cell, Organellar Electrophysiology. Just a brief introduction about Axxam. So this is a leading innovative partner research organization and IPO in assay development, high-throughput screening, and profiling and in vitro follow-up. It's founded as a spin-off from Bayer Group, but from 2001 it's independent, and is located in Italy, Milan and Naples, and in Germany. Up to now, we are 133 employees, 94 scientists, and 70% are PhD. We have offices also in Cambridge and Copenhagen. This is the expertise of our discovery services, and in particular, electrophysiology is here in the high-throughput screening and in the compound profiling and heat follow-up. The people of the electrophysiology group are PhD with several years of postdoc, and in particular, I come from the PACE lab in Milan. And our background is for sure the electrophysiology, but we have also expertise in other different techniques. Okay, we can start with the topic of this presentation. So the Axxam Organellar Electrophysiology. And the first question is why the electrophysiology in organelles? Because it's important to know that over the 80% of transport processes occur inside the cell. So Axxam developed new assays to investigate these targets. And in particular, the Axxam Organellar electrophysiology can be divided into three areas: the nuclear patch, the lyso patch, and the Mito patch, to investigate ion channels expressed respectively in nuclei, lysosomes, and mitochondria. And these targets acquired large importance in the last year, in particular because the lysosomal ion channels are involved in Parkinson's and in SARS-CoV-2, and mitochondrial ion channels are involved in cancer. Okay, the first area is the nuclear patch, so the patch clamp on nuclei. And this is our case study. We investigated the ryanodine receptor expressed on the outer membrane of nuclei. So nuclei were isolated from the cells, and we performed single channel experiments in the nuclear touch configuration. And ryanodine receptor is involved in calcium signaling that controls DNA synthesis and repair, gene transcription, and several cell functions. These are sample traces that we obtained in the nucleotide configuration, so using increasing concentration of calcium at the different voltages. And you can recognize the closed and the open states of the ryanodine receptor. These data were presented during the biophysical meeting 2020 in San Diego. Then about the lysopatch, we have a huge experience on this organelle. We investigated several ion channels, in particular TMM175, because it's associated with Parkinson's disease. And we perform biophysical characterization, permeability study, and pharmacological investigation. Then the second channel is the TPC2, 2-port channel 2. It's a very hot target in this period because it's related on SARS-CoV-2. And these channels is not endogenous in X cells, so we transfected cells with a plasmid, and the ion channels were expressed on the lysosomal membrane and not on the plasma membrane. Then we use the amitriptyline to evoke the current. Another channel is the BK, big conductance calcium activated potassium channels, involved in the regulation of phagocytosis. We tested increasing concentration of calcium to evoke the current. And then on the lysosomal membrane, several cation channels are expressed, and we perform compound profiling experiments using agonists and antagonists. Then the mitopatch, patch clamp on the mitoplasts. Mitoplasts are the mitochondria without the outer membrane. So we investigated ion channels expressed on the inner membrane. In particular, the MCU, the mitochondrial calcium uniporter, implicated in calcium homeostasis and various cancers. So the current was activated by increasing concentration of calcium and blocked by ruthenium red. On the right, we have the TASC3 potassium channel, T-resistant, related to mitochondrial morphology, membrane potential, and aldosterone production. In the mitopatch, we performed also single channel recordings using two different configurations of patch. The mito attached to investigate spontaneous single channel activity, and the inside out configuration to investigate the mitochondrial permeability transition pore. In this case, we can perfuse the calcium in the internal side of the channel to maintain the channel in the open state. Then the topic of this presentation, because we realized that in Axxam we have the assays to investigate ion channels expressed on the organelles, but these experiments are characterized by good quality of recording but a low throughput. And then the idea was to applicate the Q-patch on the Organellar electrophysiology. And the Q-patch represents the key to increase the throughput, maintaining high-quality experiments. And so the topic is the Q-patch meets organelles. It's important to know that these results are very preliminary, and I'd like to give you a few take-home messages during this presentation. So the summary of our activity, we have the Q-plate production provided from Sophion, then the mitoplast and the lysosomes preparation, the assay optimization, and the validation on specific targets. Sophion provided us organelles dedicated Q-plates. As you know, the Q-plates are characterized by substrate in silicium that permits to obtain gigaohm seal without a seal enhancer. The standard hole size is 2 megaohm in physiological solutions, and the hole size can be customized. So Sophion worked on this parameter to reduce the hole size in order to allow the organelles capture. It's really important to reduce the hole size because the organelles, the lysosomes and the mitoplasts, have capacitance from 2 to 4 picofarad. So they are very small if compared to the cell. Then the project activity plan. we started with the organelle isolation. We evaluated different isolation protocols described in literature and different commercial kits. Then the protocol were optimized. In particular, we worked on the number of cells, seeding, timing, in order to obtain high quality of organelles. The second phase is the visual inspection obtained under the microscope. So on the left, we have a cell with the enlarged lysosome inside. And the size of lysosome is increased by a chemical treatment. And then after the isolation, the plasma membrane and the other organelles are discarded from our preparation, and we have only the lysosomes. On the right, we have the mytoplasts after the isolation, in this case. And we have the mytoplast, so the mitochondria without the outer membrane, and the cap. The cap is the outer membrane that is not completely removed from the mytoplast, but remains attached in one side. And in this case, the preparation after the isolation contains several debris. And it's important to describe the results in the next slides. Then we carried out the validation using the manual patch clamp platform, where we validated our organelles by the recording of specific ion channels, and then the application on the Q-patch. We started working on the mitochondria. So we are on the Mito Q-patch. And this is the protocol that we used. It's described in literature, and it was provided from Professor Szabo from the University of Padua. And this protocol, this isolation protocol, is quite fast because it requires around 1 hour to obtain the mytoplasts. Then we move on the assay optimization. And the target is to improve the mytoplast capture and to improve the seal resistance. Because at the beginning, we didn't obtain the good capture using the Q-patch. And we work using different solution. And to make a long story short, I reported only the best condition that we obtained. So we use a pipette solution with the cesium inside, and the BAT solution at the vehicle is a physiological buffer, so this is a thyroid solution. In the bar graph on the left, we have the relative percentage of mytoplasts attached. And it's important to note that when we obtained a seal in the mytoplast, we obtained also the whole mytoplast configuration. So 100% of the mytoplasts with the seal are in the whole mito configuration. But if we look at the percentage of the mytoplasts attached, it's very, very low. And it's work in progress. We are working to increase this percentage. And the first take-home message is that the composition of pipette and BAT solution are crucial for the capture and the passage in the whole mito configuration. Then after several attempts, we were able to record the first currents using the Q-patch and mytoplasts. But at the beginning, the situation was not so good because the signal was not stable in control. The signal was saturated. We have run down, and then also leak current. So a typical APHIS experiments at the beginning of assay development. And our target at this point was to maintain signal stability during the recording and reduce the leak current in order to applicate different compounds on the same mytoplasts. And this is one on the first recordings when our conditions were improved. And we compared our recordings obtained on the Q-patch on the right with the same experiments performed at the manual patch clamp on the left. So we maintain the same solution, but we use potassium in the manual patch clamp in the internal solution and cesium and the Q-patch for the internal solution. And as you can see, the shape of the current is similar. And this is not a specific ion channels, but at the beginning we investigated the whole mitoplast conductances. And the message is that the whole mitoplast conductances can be recorded using the Q-patch. And this is the first experiments using an organelles and the Q-patch. We investigated the time dependence. In particular, we carried out experiments at different time points after the isolation. And the best results were obtained at 30 minutes after the isolation for the percentage of capture. But if you look at the seal resistance and the resistance in the whole Mito configuration, we are over the gigaohm, so the quality of experiments is very good. Then we reported an IT plot of the current. The black is the current in the control with sodium chloride. Then when we put the NMDG, we have a slow reduction for the outward current and a fast drop in the inward current. So at this point, the conclusion is that all mitoplast conductances are good for the assay development phase, but a more specific target is required for pharmacological experiments. So we decided to investigate the mitochondrial calcium uniporter because we have a huge experience at the manual patch clamp. And this is a sample traces. In black is the control and red is the current in the presence of 0.1 millimolar of calcium to evoke the current. And then we perform the same experiments using the Q-patch. The solutions are different, and as you can see, we have calcium in the bath solution, and these are the recordings. As you can see, the current in control is already active because we have calcium in the bath solution, but when we increase the calcium concentration, 0.01 and 0.1 millimolar in the bath solution, we have an increase of the signal. And then we calculated the calcium-sensitive current, and we observe a dose-dependent effect induced by increasing concentration of calcium. In parallel, we'll perform experiments on the lysosomes. So in this case, lyso Q-patch. And for the isolation, we use a commercial kit from Axxam. And this isolation procedure requires around 5 hours, so it's very slow if compared to the mitoplast isolation. We maintain the same scheme of experiments. So at the beginning, the target was to improve the lysosome capture and the seal resistance. Also in this case, we use cesium in the internal solution, and we use 2 different solutions for the bath and the vehicle. And as observed for the mitoplasts, also for the lysosomes, when we obtain the seal, we have the passage in the whole lysosome configuration. In this case, the percentage of lysosome attached is higher if compared to the mitoplast. And in particular, we have a good percentage 90 minutes after the isolation. And also in this case, the first conclusion is that the composition of pipette and bath solution are crucial for the capture and the passage in the whole lyso configuration. Then we started to perform experiments and to record currents in lysosomes using the Q-patch. The situation was quite good at the beginning. We worked on the application protocol, so to increase the timing of the liquid period in vehicle. We sometimes observed run up and run down. But then we moved on a specific lysosomal target, the TMM175. We performed a lot of experiments at the manual patch clamp on this target, and in this case I reported the different solution used in the bath solution, so potassium and NMDG to reduce the current. And we performed similar experiments using the Q-patch, but not with different solution, but with a non-specific blocker, the zinc. And as you can see, the black trace is the current in the control. Then we put an increasing concentration of zinc, and we observe a dose-dependent reduction of the current. So in this case, the take-home message is that a specific lysosomal target, in this case the TMM175, can be investigated using the Q-patch. The time dependence for the lysosomes. So we carried out experiments at different time points after the isolation. Best results were obtained 90 minutes after, and also in this case the seal resistance and the resistance in the whole cell configuration are over the gigaohm. Then the IT plot of TMM175 current. Black is the control, the light red is zinc 0.01 millimolar, and dark red is 0.1 millimolar of zinc. And on the right, the traces with the dose-dependent reduction induced by zinc. So we observe TMM175 dose-dependent inhibition induced by zinc. But this is not a specific blocker, and we would like to repeat these experiments using, for example, the 4-aminopyridine. So to conclude, we have general conclusions. We are able to obtain a higher number of lysosome capture than the mytoplast, but it's due to the purify, so the debris in the organelles preparation. Up to now, Q-patch permits to obtain similar daily data points compared to the manual patch clamp, but if we increase the percentage of capture, the success rate of Q-patch will be much higher than the manual patch clamp. About the Mito patch, preliminary results on the MCU, mitochondrial calcium reporter, are really promising. And also for the lyso Q patch, we obtained the promising results on TMM175. But additional experiments are required to perform permeability and pharmacological characterization. So the last take-home message is that the Q-patch is suitable for Organellar electrophysiology on both mytoplasts and lysosomes using dedicated Q-plates. And before to conclude, I'd like to thank Axxam, and in particular Jean-François Hollande, Elias Carabotolo, and our CEO Stefan Lohmer for the support in this project, and also Sofyan for the opportunity to work on these targets and to present this data. Thank you.
That's great work, Manuel. Thanks for the talk. It's really fascinating stuff. All this Organella ion channel recordings. I mean, one thing that strikes me about it, and we've obviously had some discussions prior to this, but I mean, it seems like manually it's actually really, really technically challenging. So to be able to sort of do it in automated patch clamp is really a phenomenal sort of achievement that you're trying to do there. So it's looking really promising. I guess one thing that sort of occurs to me off the bat is potentially an easier way is to have these cloned Organella ion channels and potentially express them in the plasma membrane. But that ease may come at some cost. What are the pros and cons, do you think, briefly? I don't want to sort of hog this spell now, but what do you think the pros and cons between expressing in the plasma membrane an Organella ion channel versus going straight to the Organella ion channel recordings in the organelle environment as you are trying to do at Axxam?
Yes, the possibility to express the ion channels, lysosomal ion channels on the plasma membrane is a good idea. And we performed these kind of experiments because it's faster than the lysosomal patch clamp. But the environment and the composition of plasma membrane is different if we compare that to the composition of lysosomal membrane. And so the idea is that a primary screening on plasma membrane is good, but for a complete validation of drugs, that can act on specific lysosomal ion channels, the lysopatch is the more physiological system, and it's required.
Of course, yeah. But yeah, hats off to you. It looks fantastic work and very challenging, but it's great that you're getting some really interesting results already. We've got a question here from Professor Jonathan Ashmore from UCL. Any idea why the currents in the Mito patch are apparently larger than when you use a single patch pipette?
Yes, we observed a larger current in the Q patch, and the trace that I presented was one of the first recordings that we carried out at the Q patch, and probably some leak current were present during that recordings, and the current amplitude was reduced during the last experiments, and at the moment the current amplitude is compared from Q patch and manual patch clamp system.
And just to follow on from that, do you think that potentially, I don't know, would it cause problems, or is that just a case of that's what you see and it happens to be bigger, but it won't necessarily affect the biophysics or the pharmacology because of that larger size?
Yes, probably we have also different composition in the external solution, in the batch solution, and it modified the expression of these channels, and then also the different technical experiments in the Q patch and in manual patch can modify the expression of these channels. So during the manual patch clamp, we have a continuous perfusion, and in the Q patch is not present. So there is a different stress of the channels, and it can modify the expression.
Okay. Just another sort of call to anyone who wants to ask some questions. Of course, this can be carried on in the breakout sessions at the very end of all of the talks. But if you, last chance to get your question in now. Otherwise, you can catch hopefully Manuel in the breakout sessions afterwards.